So as a result of that, it will grow in an exponential manner. So for example, see, theoretically, I'm not telling you practically, after 30 cycle, you will
Sorry
get...
to disturb, sir, but somebody has stopped recording.
Oh, 1 minute. But here it's showing recording is going on actually. So recording is going on, okay? So no problem.
Okay, sir.
Yeah.
So here they are given 3 cycles, that temperature division for 3 cycles, but my question is like why like we cool down the strands in 1 cycle only, right? Like we do all the 3 in 1 cycle only.
I got your question. So the thing is, you can ignore this cycle 1 and cycle 3. That is, I would say, not required for your understanding purpose or anything. So the cycle I mentioned here, these 3 actually, that 98 here mentioned, you can do at 94 degrees centigrade also. The tube is like, very high now, that in the temperature of the tube. So that strands are separated. Then you are cooling down, primer will be annealed. And then at this temperature, like the strand will be synthesized. Now from 1 DNA, now you have 2 DNA. Then again, it will come back here. Okay. So this one you are doing in cyclical manner, like around say 30 to 35 cycle. And then here, this is for different purpose. Even if you ignore this part, it should be fine. Because some of the portion, if the synthesis is not full, then it will be completed here. Like those things are there. So it is fine if you ignore this part. But this is the major part here, how like 3 different temperature help to synthesize DNA in presence of Taq polymerase, that is thermostable DNA polymerase, and you can amplify your DNA. And now, like as you can see, 2 to the power 30 means this many numbers, so that many copies of DNA can be synthesized within short period of time. And now, when actually scientists first invented this procedure, what at the very beginning, what they used to do, they have 3 defined water bath that set 3 defined temperature, and they have one tube like this, and they are putting this tube in like 94 degrees centigrade temperature, waiting for 1 minute, then like little low temperature, then again 72, because 72-degree temperature is very like optimum for this enzyme function. So but now we have, because of this, I would say like physics, like physics and other technological improvement. So we have good equipment like this. So this is very common. We called it thermal cycler. This one, like you can see here, or like commonly you can say PCR machine, but it is actually thermal cycler. So this is here, it's kind of very small machine here, and you can see that here actually small holes are there. So you can put this kind of tube, like small tube. The like maximum amount it can contain like 100 microliter or something like that, very small tube. So you can even put 96 tube together in this plate, and this machine will help you to like keep this temperature, like every cycle it will go up, down, and again at 72-degree, and you can adjust your temperature whatever you want based on your reaction, whatever you want to do. So this is called thermal cycler, and this is common in any molecular biology lab. And also the similar machine is being used for like RT-PCR, PCR, all those kind of thing. Okay, so this is about polymerase chain reaction, and I have one more slide here, I believe, about DNA analysis in forensic science. As I told you, in order to analyze the DNA, you have to increase the amount of DNA first. And like although in academic lab, this is very common, almost every day we use PCR for different reasons, but in forensic science, it is also very important. And as you can see, there are different methods, and this DNA analysis is also one important method actually. And here you can actually establish the identity of a person. So that's all about like DNA sequence, not DNA cis polymerase chain reaction, but in order to establish the identity, you have to do the next step, that is DNA sequencing. I am going to discuss now.
So here we know the sequence, like we know the end sequence of the DNA, because how are we creating the primers then?
Yes, so I expected this question. So very nice, you thought about it. So I was expecting that someone will ask. That's why around 20 minutes back, 20 to 30 minutes back, I told you that human genome sequence is known, right? And I gave example that my sequence and your sequence are very similar. Only you will get like find like one or two nucleotide. If like you sequence my DNA and your DNA, like for example 1,000 nucleotide long, like at a stretch you are sequencing it, you will find only hardly 2-3 changes there. I'm just giving example. So, and those primers are small actually, right? Like 12 nucleotide, 18 nucleotide. So as a result of that, when we have like some sample, say for example, like any body fluid, like one drop of body fluid, and we want to actually extract the DNA. So we have good chemistry now in order to extract the DNA from that body fluid, like from blood sample or whatever it is. So we can extract the DNA. Now we are expecting that we have like the whole DNA there, like maybe minor, like little amount. Now, since we have the genome sequence, that means we know like what is the sequence of human DNA overall. It will be little bit difference between like X and Y, like person like person A and person B. But still, now we can use some set of primers, like what you asked, that how we will do that. So since we know the human genome sequence, we will use some specific primer and then we will use that. Maybe in accidental case, one primer might not work because maybe in that region you have a lot of differences. Like accidentally it might happen, but still we can use another set of primer. That way, the goal is like, what we do, like in forensic lab, what they do, like 3 to 4 like different gene they want to amplify. That 3 to 4 different segment they actually amplify and then they sequence it. And if you do that, like 3 or 4 different segment of DNA you sequence, like I would say 500 to 1,000 nucleotide sequence segment of DNA at 3-4 different location, you sequence like my DNA and someone of your DNA, you will get some differences. From there, you can establish the identity. So is it clear, Tanmay?
Yes, sir. And just one more question. So the primers in case of DNA replication, we saw that they were RNA, small RNA segments, and later at the end of replication, they were in the video they showed that they were removed, like there was an enzyme. So do we do the same in PCR also?
No, PCR is DNA primer because we are doing in vitro inside the tube, right? Why we will add RNA primer? That will be more problem for us, right? So because these primers are commercially, like you can buy it from some company, or if you have good biology and chemistry lab together, so some of my chemistry friends, they can make primer for my, like, for my lab, right? So we are synthesizing this oligonucleotide in the lab. So as a result of that, we synthesized the primer. Here the primer means DNA primer because we are making it in the lab, right? Through some chemical reaction procedure only, not through biological like replication procedure. Just primer means you have like, for example, I have provided like GATC sequence to make the primer. So in chemistry lab, what they do, they have like some solid phase synthesis. So you have G, then you have to add A. So that time they are flowing only A, right? So that way you can incorporate A residue. Then next one, and then you have some quality control mechanism, and then you will provide the primer. So this primer are DNA primer. Okay.
Yes, sir. So
Yeah.
we have a set of DNA primers already available for human genome.
Yeah. For this purpose, and if you want to make some new primer, you can order. Like you can synthesize or you can order also because the genome sequence is available now. That's why.
Okay, sir. Got it.
Okay, so just one thing. I hope that like all of you can understand this thing because I'm going slowly and I'm discussing with student, like whoever asking question, but I believe the similar questions you also have in your mind. Like maybe you are not discussing and like all of you, like all 100 of you discuss that way also, I cannot like go forward. But I hope that whatever we have discussed, those are pertinent questions and which need to be clear and you are fine now. So now let me move into sequencing part. This will take around, I would say, 30 minutes again, and then you will take break. DNA sequencing is very important. Like I would say important means not for exam, I'm saying very interesting. And also you need to understand this PCR thing very nicely and the replication as well as the agarose gel run, the overall the DNA gel electrophoresis. Then it will be easy. Like if you know all these three steps, then it will be easy to understand how we conduct DNA sequencing. Again, I want to tell you that DNA sequencing also very common, right, in molecular biology lab. Even during my own PhD, I used to sequence DNA in a very regular basis. Long time back, I'm telling you, almost like 15 years back, 15 to 20 years back. So let me share my experience about DNA sequencing, how we conduct DNA sequencing. Now there are a lot of improvement because of very high level of technology, but the basic mechanism, the reaction mechanism is the same. Whatever technology we use, but the main principle is same. How we sequence DNA in today's world. I will go with the whiteboard completely now, and then you will get this slide. DNA sequencing. One great scientist, actually, many of you know, if I ask you that who got Nobel Prize twice, many of you will answer, I'm sure. And I believe that none of you will answer this name, actually. That's why I'm writing Frederick Sanger. So he got Nobel Prize twice, right, in chemistry, both the time in chemistry. First time he actually sequenced and provided the structure of protein, insulin particularly, and in 1958 he got Nobel Prize. You don't need to remember name as well as year, nothing. I'm just telling you 1958. And then in I believe in 80, again he got Nobel Prize for DNA sequencing. See, I told you that there are like few major like macromolecules in biology in living system like protein and DNA, RNA, carbohydrate, lipid. And this guy, like this scientist Sanger, he sequenced and provided a lot of information about both protein as well as DNA sequencing. And for both the time, he got Nobel Prize. So whatever we do even today, the basic principle is whatever developed by Frederick Sanger. That is also called Sanger sequencing. So let me discuss the process. So during DNA sequencing, as you know, that DNA, both strands are complementary. If you know the sequence of one strand, then the other strand, you can automatically write the sequence of the other strand. So as a result of that, during DNA sequencing, we will concentrate on just one strand because your job is to sequence just one strand. That is done, right? So from this point, that's why we are concentrating on just one strand. Then the other strand is automatically you will get it. So let me tell you. So here, one DNA present here. I'm just writing, some kind of sequence. This is a DNA strand. The other strand I am not writing here. And also remember that whatever sequence I have written here, that this is just to explain you, but you don't know the sequence of this full DNA, right? Without writing, I cannot explain the procedure. That's why I wrote. Now, if this is the DNA sequence, we have to derive this sequence, whatever written here, whatever the nucleotide sequence here. And that is the way how we can sequence someone gene or genome, and we have to establish the identity. Or during cloning, regular cloning procedure in lab, we clone a lot of stuff. So during that time also we have to sequence DNA, like sometime bacterial DNA, sometime viral DNA. It is like going on in a routine basis. So the first thing is here also we take advantage of some primer and we synthesize the DNA. And at the very beginning, you will not understand every point what I'm talking. Let me talk about 5 minutes and then you will ask me question. So the first thing, we will use some primer, say 5' here, the primer sequence here. So T, A, C, C, G, G, C, G. Say for example, whatever I have written here, this is the DNA what we need to sequence. We have many genes, like in human we have say around 20,000 genes, protein coding genes, 20,000. So we are interested like few of those we want to sequence. And as a result of that, just like PCR, like someone asked me that what segment we will amplify. Similarly here, we will use some set of primer because human genome sequence we know, but we will sequence a long stretch in order to find out even the small difference between me and someone else. Or like here in this case, I would say just we want to know the sequence, whatever written here. Like, so as a result of that, we use some primer and that primer will be annealed here. So this is primer here. Okay, this is primer. And this will be annealed with the template stand. That means hydrogen bonding. If you know the sequence, then it's fine. You can use some specific primer. If you don't know the sequence, then what will happen? I'm sure that someone will ask this question. Like, for example, you have discovered some new organism, some new virus, some new bacteria. Even the sequence of that new virus or bacteria is not available. Then how you will sequence? You have to sequence it, right? So I would say in that time, what we use, those primers are small. So we use some kind of random primer. Random primer means like in terms of permutation combination, like we synthesize a lot of different types of sequence. And whatever the sequence that will be preferred, that means that will anneal to the template, that will be annealed and will be extended. Okay, so right now don't go into that complexity right now. Better I should concentrate on that some specific individual, like we want to sequence some human gene, right? So how we can do that? That I'm going to explain. So in this case, what happened? The primer will be annealed to the template sequence in proper condition. That means proper temperature. During sequencing also, you need DNA polymerase, you need DNTP, you need magnesium ion, but something also special thing that you never use in case of replication or in case of, I would say, polymerase chain reaction. And that special thing actually discovered by Frederick Sanger, and that's why he got Nobel Prize. So what I would say, so you can carry out this reaction, this sequencing reaction. This is just like polymerase chain reaction. Actually, that means you are synthesizing the DNA, not polymerase chain reaction, but you are synthesizing the DNA. So while you are doing this, so for example, you have this template here, right? So you have template, you have primer, and you have DNT base. How many? 4. That's why I put S here. DNT base, ATGC, all are there. You have magnesium ion and enzyme. all thing I told you. The reaction is happening here. So as a result of this, if this strand is being extended, then what will be the result? Let me write the result. That means if this strand, that means this primer is being extended by this enzyme, then what will happen? So it will be like 5 prime T A C C G G C G. This is just like the primer. Okay, the same primer. The primer will be extended. Now you have A in the template strand. Then what will come here? Tell me.
T.
T. Very good. So T will come here. Okay. T. Now, very interesting thing here that in this tube, whatever I told you in this tube, whatever ingredient present, in addition to that, you have one thymine residue T that is defective. So this star means T star means the some kind of defect present in this T. Again, I'm telling you that you have template, primer, DNTPs, that means all ATGC, magnesium, enzyme. In addition to that, you have some defective T in this tube. So as a result of that, what will happen? That whenever this defective T is incorporated, the strand will not be extended anymore. Some
So
kind
can
of
you please
defects.
repeat this part because I was actually not able to hear properly.
Yeah. I will repeat like every step I will repeat the same thing, so you will get it. So in the tube, you have all sort of reagent, whatever required, like template, primers, DNT pits, that means ATGC, magnesium, and enzyme. So as a result of that, the strand will be extended like TAACCGGCG. This is the primer sequence. Then if I write the sequence, like, if we continue here, what will happen? If you don't have this defective T, then the result will be AT, like A, here T is incorporated. Then you have another A, another C, another G. It will be continued like this. But we are not interested. This is happening in case of PCR, polymerase chain reaction. We are making the whole DNA. But here we want to sequence the DNA. We want to know what is present here in terms of nucleotide sequence. So as a result of that, we have to make some kind of tricks here. So in order to do that, we added some defective T, some kind of defect there in the thymine residue, defective T. Whenever this defective T is incorporated or added in this growing chain, T star, the defect, then this strand will not be synthesized anymore. It will stop. It will stop the reaction. Okay? Up to this is clear. And then what will happen? I would say in the same tube, another product might be, because the template strand, you don't have just one, you have multiple template strand, right? So another option might be TAACCGGCG. This is the primer. Okay, this is the primer. Then instead of the defective T, the normal T, because you have ATGC, normal ATGC also present. So if the normal T incorporated, the normal T added, then this chain will be extended, right? So normal T here. So as a result of that, the next residue is T here in the template. Then what will come here? Tell me.
A.
Then
C, G, C.
G. Then again T. Say, just think again, in this position you got this defect here. Okay. Then again, another reaction might be, you have the primer sequence TAACCGGCG, and then TACGT. So here again one normal T actually incorporated. So as a result of that, it will be extended, and when another T should be incorporated, say for example here you have another A. So as a result of that, again at some point later another tube will come. So here the T star, that means defective. So here get it clear that in this tube, whatever product you are getting, all of those products are done by, or at the end you have one defective T or defective thymine residue. And then you can ask me now, sir, then why it will be extended? Because at the very beginning, if all the defective T are added here, then it will not be extended. My answer is yes, you are absolutely right. But in order to sequence the whole DNA, we want the reaction should be continued, right? In order to do that, what we do here in this tube, whatever you have the, here you have normal ATGC in terms of DNTP, like normal A, T, G, C, all are there, and also defective T. The concentration of defective T and normal T, I would say T is to T star. You can say 99 is to 1, something like that, 100 is to 1. That means you have mostly normal T, only very little amount of defective T. Otherwise, what will happen if you have all of them are defective T? So at the very beginning, all those defective T will be incorporated or added, and then the chain will not be extended. But our goal is to sequence the whole DNA. Like I just wrote here few nucleotide, but you have like many things here. So you have to sequence. So you have to allow the DNA to be synthesized. So that's why we put like 99 is to 1 or 100 is to 1 in that ratio, so that the defective residues are much smaller in number, so that finally what we are getting in the tube A or tube 1 here, in tube 1 we will get different length product, and all of them they are done with the defective T at their 3' end. Is it clear or not? Rajas, is this clear?
Yes, sir.
Okay, but the story is not completed. I think that you are not getting the point yet, but if I like continue another few minutes, then you will get it. So now this is very important. Just try to understand properly. Now in the next tube, what happened? So this is just tube 1. In the next tube, what we can do? We will do the exactly same thing, but instead of defective T, we will put defective A. Okay, defective A, and other things are everything same. So then what will get? So every product will be done with what? Tell me.
A star.
Yeah, so at the end, like every product will be done with defective A, because here we added the defective A. So this is your tube number 2. And here tube number 3, we will put defective C. So every product, whatever we will get, those are done with this defective C residue. And the last tube, because we have only 4 sequence, like 4 different nucleotide, ATGC. So here we will put defective G. So as a result of that, this is 4th tube. So we will get all product that are done with defective G. So now the thing is, what I'm trying to say, whatever the sequence present here, right? You have all different length product. The only thing that product present in this first tube, they are done with the T at the end. Here in the tube 2, they are done with the A, but you have all sort of combination, because you are synthesizing those things in 4 different tubes. And now what you will do, you have to run one gel. I'm not going into like very details of that gel, but I want to just explain in this way. So if you are running one long gel, so gel means again you have to put some negative electrode here, here positive electrode, and here you will load your product from tube 1, then tube 2, then tube 3, and then tube 4. Okay. So you have loaded your sample here. So now, if you run the gel, the DNA is negatively charged, so it will go from negative to positive direction. So can you tell me that from like DNA will be separated based on their mass or their size. So if I tell you that where you will get the smallest product, in which tube, tell me. Again, I am asking the question, in which tube, like that 1, 2, 3, 4, in which tube you will get the product which is smallest in size?
Sir, with regard to this given DNA sequence,
Yeah, given sequence, right. Whatever I was explaining here. Yeah.
sir, then it will be 1, I think, because at start it is only T.
Very good. Yeah, the tube 1, right? Because here, if you see, because whatever sequence I have provided, that it will stop like at the first T. So as a result of that, you will get the smallest product in tube 1, and the smallest product migrate faster. So after the gel done, you are getting one band here because of this smallest thing here. Then another band you will get in which tube? Because the next one is C, the done with defective A, right? Here, like it is not done, but if you like see the different length product in tube 2, you will get here you have defective A. So as a result of that, since you have this A then T, then the defective A here, then here, then here. So based on their size, because all these DNA strand, they are separated, or their difference is just one nucleotide, because all sort of combination actually synthesized in these 4 tubes, and you are loading those product in 4 different length, and as a result of that you are getting like this. And I put this sequence like this ATGC, that's why this pattern is coming, but it can be anything, but based on their size, you can deduce the sequence. Now imagine that you don't know the sequence here, you don't know, but only thing you have carried out this reaction. And if you get this product is the smallest, that means primer plus just one nucleotide. So then in the first tube, you know that you added defective T. So then after primer, what you have? You have T residue. This is the smallest product. And the next one, 5 prime, then T from this lane 1 or tube 1, you already know that this is the T. And then the next one in tube 2, you have the A residue. So TA. And next one, 5 prime, TA, this one, 3, you have C, defective T, TAC. So that way you can actually deduce the sequence of this DNA. So as
we have many T star here, like the smallest one is also there, the a bit larger than that also there. So we have to look for the one that has gone the most distance. So
yeah, no. So you are saying many T star, but that's why if you run this gel, long gel we use, so all T star will be like arranged like this, right? Right. So this is the, I would say, very high, like very bigger length. This is the smallest length. Is it clear?
Yes, sir. That means we have to look for the one that has gone the most distance. So
yeah. And then you have to start start calculating what is the sequence. Okay. So that way we do. So I will show you the slide, and whatever remaining question you have, I'm sure that slowly it will be clear. This is just just the like mechanism I explained. First, give me one question here to me, that I intentionally told this thing that defective T, defective A, that way. Can you tell me that what is the defect here? Because of this defect, he got Nobel Prize, right? But what is the defect here? I didn't tell you the answer yet.
is it RNA? By any chance?
No, this is, I'm telling you that the nucleotide defective T, defective A, but can you guess what is the defect there in the nucleotide?
Sir, is it
Sir,
radioactive?
polyester
Sir,
is
it does not
related to that.
contain
Radioactive.
OH group, another OH group.
Yes, sir.
So Razan, Reva, so you are trying something. Yeah.
Yes, sir. Sir, it is dideoxynucleotide triphosphate.
Yes, very good. The thing is, this is very important. Whatever you told right now, I'm just telling you that all nucleotide sequence, like whatever the nucleotide you know that you have here, you have the A, T, G, C, whatever you have like nucleotide here. This is the pentose sugar, 1 prime, 2 prime, 3 prime, 4 prime, and then CH2 OH. And you have phosphate here, right? Phosphate, if it is triphosphate. And here you have H and H in case of deoxyribonucleotide, in case of DNTP. And here you have H and OH, right? So this is 3' OH, and this 3' OH actually that helps in that nucleophilic attack, and so that the chain can be elongated, as I discussed during the application, right? But here the defect is nothing but whatever she told, that this is called dideoxynucleotide, just one derivative of this nucleotide, just H. So as a result of that, this is now ester that I put here adenine residue. That's why ester. So whenever this nucleotide will be added here, like if you see this is not A, this is T. Whenever this T will be incorporated, then what will happen? Then you don't have OH, you have just AH. So as a result of that, the next nucleotide cannot be added because you don't have the OH, which is required for the next reaction. So this is now I'm telling you the T star or the defective T. This is called dideoxyribonucleotides or DDATP. Just I'm just telling TTP here, thymine, dideoxy. So actually, Sanger was he was a chemist. So he formulated this thing. He designed this nucleotide, and he actually cleverly used this nucleotide in order to get all sort of fragment, whatever possible. And he was carrying this reaction in 4 different tubes. Now I believe it will be clear that whatever I explained, that here like 4 different tube, and they have like in this, if you mix like all different like defective T, A, C, G at the same tube, then it will be problem here. That's why he carried out this reaction in 4 different tubes. So as a result of that, you know that in channel 1 or in lane 1, whatever the product you are getting, they are done, or they are at their 3' end, you have the T residue, because in tube 1 you have only defective T. That way he cleverly designed this experiment, and he was doing this thing. So let me go through the slide, then I will take your question. So whatever I discussed, everything is mentioned here. The only thing I didn't tell you that they label the primer with some radioactive, because the amount, the product you are getting is very little, but if you have some radioactive signal, you can track it better after running the gel. I will come into that that what's the advantage or disadvantage of this radioactive thing. But nowadays in lab, whatever we have used, that is non-radioactive method. Okay, but right now you can forget this thing. Without knowing this, it is fine. You can get the major point here. So here, this is normal DNTP. You have OH, and this is DDNTP. You have the H only here. So as a result of that, whenever this nucleotide will be incorporated, then it will not be extended because of this absence of this OH group here. And this is the defect, and this is being used. And whatever I discuss, everything clearly mentioned here. This is the mechanism how OH group is required, and then whatever I tried to explain you in whiteboard, you will get the similar explanation here. And I told you that almost 100-fold lower in concentration, like the defective one you should use in a particular tube. That is also mentioned here. So as a result of that, finally, if you have this kind of sequence, this is the template sequence and this is the primer. And if you are making this, if you're making this reaction in 4 different tube and that contain like dideoxy-T, dideoxy-C, dideoxy-guanosine, and dideoxythymine, then you will get like all sort of product. And if you run in gel, and this is the reaction is not mentioned here. Like this is the negative and this is the positive. So as a result of that, here you have the smallest product. So you can see this is starting, the start GNA is 5 prime G. Then you have this one A. So instead of tube 1, 2, 3, 4, here it's mentioned that channel 1 means you have the defective A in this lane. So then the next residue or next nucleotide will be A. Then here T, then another T, see, another T, then this one C. So that way you have to continue. So you have to read this sequence and then finally you will get the nucleotide sequence, whatever you want to sequence. Is it clear? And if you have some question, quickly ask me.
So can you explain that radioactive part once again? I did not quite get it.
I didn't discuss actually, Aditya. So I will come into that. Right? I didn't like I just told you, just ignore for the time being. But I will be very happy if you understand the other portion, whatever I discussed, the major understanding behind the sequencing. I think it is fine now. Tanmay, is it clear?
Yes, sir, got it. We have to check the levels of the gel electrophoresis in order to determine the like how much it has gone based on that.
Yeah. So I will so I will like now I will little bit emphasize on the radioactive thing as well as modern day what we do. Normally we want to avoid the radioactive thing like in the lab. So that's why I will tell you little bit improvised version of this strategy what we are using nowadays in the lab. But the basic chemistry is same. The Sanger dideoxy. This is called Sanger's dideoxy sequencing. You can understand from the name.
Excuse me, sir.
Yeah.
Sir, but means for one nucleotide we can identify that is A, T, G, C. But if it has 2 or 3, how do we know that it's like next one?
2 or 3
in the step.
like consecutive A, you are saying?
No, sir. I mean, we can identify which one is the lowest one. I mean, in the gel electrophoresis, like AGCT if it is single. But if there are like 2 or 3, how do we know that it is next to that our sequence?
So if I get your point correctly, I would say like, since all like, all these 4 tubes, whatever you are getting, like all the product you are getting there, if you like make some kind of combination, you will get like throughout the length, you have all sort of combination. Only thing is that in tube 1, you have all thing ended with, say for example, you are getting this is tube 1 where you have T. And then maybe the next tube G, you are getting here G, A, C. So you are getting 1 here. So that means the sequence will be T, T and G. You will never get like in the same migration or same distance, you will never get like this. Then something wrong because you have just one type of DNA template here. So how at the same position you will have 2 nucleotides? It is not possible, right? So you will never get like something like this, because then you are done. Like you cannot tell whatever is present here. So because at one position you have just 1 nucleotide. So as a result of that, this situation will never arise. So as a result of that, always you will see that those like chain or those product are at least their migration is little bit different, and based on that you have to determine that what is the sequence. Is it clear?
Yes, sir.
Yeah. So now,
here we are saying that we might need to use some labels Sir, in gel electrophoresis, we add some stain like ethylium bromide and we observe it under UV light. So why do we need labels in
Yeah,
order
yeah,
to
yeah.
understand?
Yeah. So, Tanmay, so to everyone actually, someone like they are asking question, but I believe you have this question. So, in gel electrophoresis, we just want to. This is I would that gel electrophoresis I would say is kind of crude. You cannot really differentiate between one nucleotide difference. There you have like some DNA which is 50 nucleotide long. Another one is like 75 nucleotide long. That way you can differentiate. But here this is much more sensitive. Here just one nucleotide difference, still you want to differentiate. Otherwise you cannot read the sequence. Just one nucleotide difference, you have to read it. Like number one. Number two is also in PCR, you have the same length product in huge amount. What you can visualize just with your naked eye if you add some ethidium bromide or some other derivative, some like nowadays even ethidium bromide is toxic to us. We use something like more friendly reagent. But in this case, the product is also less because this is not like PCR. And also we want to read almost every length product, all like, like just they are difference by one nucleotide. So as a result of that, you have to have some kind of more sensitive thing. What like traditionally people used to do, Sanger, what he has done, that those primers, they are labeled with some radioactive phosphate, right? The phosphorus is radioactive in the primer itself. Just one phosphorus is radioactive. We have like I'm not going into that details that how we can label that primer. That 5' end you can label with one radioactive phosphorus. I'm not going into that. But if you have the radioactive incorporated with the primer, then whatever the gel you are getting, say for example, like the gel is very long. Say for example, this is a white paper, but consider this is a gel, right? And now in the gel, what happened? So 1 minute, I'm writing something. So in the gel, you have this kind of band, but this band you cannot see. DNA migrated. Here you don't have ethidium bromide, so you cannot see it. But all those primer, they contain some radioactive signal. So after running the gel, we dry the gel. The gel is dried. Then on top of gel, we put some some autorad plate. That means I'm not sure whether you guys are aware about like developing like traditional film. Like nowadays you take your photograph in digital like version, like in mobile phone, digital camera. But I'm sure that you are aware that previously you used to take some kind of like traditional camera with some film, and then we develop that. Similarly, in this case, what happened? We put some X-ray plate, right? And there the radioactive signals are coming, and then we develop that. So as a result of that, although you have small amount of sample, but because of this radioactive signal, that will create that band after like some developing steps, right? So as I told you, like almost 15-20 years back, I did this thing, but during that time I used to use this this radioactive thing. But now whatever my students are doing, they are not using any radioactive thing. They are doing something more modernized thing. I will come into that right now, that how you can avoid this radioactive thing. So here the thing is, I would say very simple, how you can avoid the radioactive signal or radioactive phosphorus here. So here I'm just deleting some of the portion here, otherwise I cannot explain. Yeah. So the thing is, so now what we are doing? We are doing this reaction, this Sanger dideoxy sequencing reaction in just one tube. Okay, just one tube. Instead of 4 tube, you have just one tube. Whatever reaction, like A, T, G, C, everything there, and instead of that defective A, defective T in different tube, what we do? We have all this defective ATGC, that means dideoxy ATGC, in the same tube. But the difference is you have A, T, G, C, all these 4 nucleotide with 4 different color. That means those dideoxynucleotide, they are tagged with some fluorophore dye. Say for example, the A with the green color, some fluorescent dye, the T with red, and the G with blue, and the C with black. Is it clear that we are adding dideoxy ATGC in the same tube, but all those 4 nucleotides are differently colored. That means in addition to the nucleotide sequence at right position where the reaction, where if you add something, the reaction will not be hampered, but you have some fluorophore dye, 4 different color. So as a result of that, when you are carrying this reaction in one tube, then how you will analyze this thing? So here you don't have radioactive thing, so it is much simpler, but you have to have some good technique for that. So what we do after the reaction, we load that product in, it should be straight line, okay? So we load that product in some long capillary tube. So reaction is over, so we loaded here sample. This is called capillary tube. It has some, it contains some gel kind of matrix, right? So that the DNA will be separated. And in the capillary tube also, you can put some electrical field. Again, you have minus and here you have plus. And the product will be separated inside the capillary tube. So again, you have negative and positive here. So as a result of that, the smallest fragment will come faster and the biggest fragment will come later. So as a result of that, when the product, like when this DNA fragment, they are coming out from the tube, so they will be separated inside the tube based on their size. So for example, if you see here something like red, that means it should be T, because I have attached the red fluorophore dye with T. Then maybe another T, another red band here. Then maybe one A, one green here, one G, one blue here. Then how you will distinguish this thing? So this is today's sophisticated thing, that in the capillary tube, you loaded this thing, and then while it is coming out from the capillary tube, you have some laser and you have here some detector. Okay, detector. And now if you plot this thing, so you will get, so you have like the first, this red signal will come, so you will get something like this. And then another red thing will come, then another signal like this. The detector will detect. Then what you have? You have green. Green. Then what you have? You have blue. For example, you have black then. Then what you will get on black? Then again another, for example, red. Red. So if you get this kind of chromatogram, this is called chromatogram, because they are showing different color chromatogram. So here you have, from this chromatogram you can tell, because you know that the red fluorophore dye is attached with your T. So the sequence is T, T, then the green is attached with A, and then the blue with G, then the black with C, then the red with T. So that way you can read the sequence. So this is today's sequencing, how we do right now. Okay, so tell me if you have any question. I hope that you get the point. Is it clear?
Sir, just wanted to confirm that the color is applied on the defective ones, no?
Yeah, defective one, only the defective, right? That's why that defective will be added at the 3' end, and that is done. So as a result of that, that one DNA stress that should not contain 2 color, just 1 color, because you have 4 defects and 4 different color attached to that defects. Is it clear? So it is much more simple and it is very fast also. Like very quickly we can do it in the lab, right? So that way we sequence DNA currently. This is called automated Sanger DNA sequencing, not like radioactive level, no gel run, like, you know, not those like complicated things are required. This is much simpler and much user friendly also. Right, that way we sequence our DNA. And now if I want to see what is present here, whatever I told you, you will get the similar thing. I just try to explain in board, and then whenever you will get those things during preparation, you can go through your slide. And whatever I told, I'm sure that everything will get in the slide. And beside that, I also explain some extra thing. And this is the textbook, mostly like any textbook you can read. Like if you are interested and if you have some problem in some portion. Otherwise, if you can get the point what I'm discussing, like that should be sufficient also. For
This method of automated sequencing is faster than gel electrophoresis, the one that
much faster, because the problem is, see, in gel electrophoresis, the limitation is the length of the gel, because whenever the gel run is over, you have taken the gel and then you dry it and you put that that X-ray plate. The problem is that some portion will be separated nicely, but you have to run again to see the bigger product which will be separated. It is like multiple gel you have to run, and it's time consuming. But in this case, just one capillary tube, you are just waiting, and based on the mass, their sequence, they will be separated. So as a result of that, this is much faster, and I would say even less costly also, because running the gel and using a lot of things, it is more costly also. But nowadays, I believe no one actually, even I didn't see anyone that they are using radioactive, that previous technique. But that is what discovered by
just related to this, only one question I had, like in the beginning of the pandemic, they in the Chinese labs, they actually detected the sequence and they spread it to the entire world. They did it like this only, in this method only.
So nowadays, whoever sequence, like, so the DNA sequencing is whatever I told the last portion. So this is the common thing. Now the thing is, see, if you go to if you see at IIT Kharagpur, say for example in our lab, whatever we have, not like this automated sequencing, we have central facility actually. So there we have some machine that contain one capillary tube or 4 capillary tube, and that laser can detect only from coming from 1 or 2 capillary tube. But if you go to some very good institute like MIT, for example, or any other good institute, so you will get maybe at the same time 96 capillary tube they are working, and they have good detection system. So that way they improve the thing. But the overall mechanism is same actually. The way the sequence, the basic, the fundamental thing involved here, whatever I discussed, that is same. But then based on the technology, how you improve this thing, right? So I'm just since you asked this question, I would say when this automated sequencing and its improved version came, because of that, human genome sequencing. In the next class, in the next topic, when I will discuss about human genome sequencing project. So during that time, since human genome is too big, it is almost impossible to run this kind of radioactive gel and derive the sequence of whole human genome. We can sequence a portion of a gene, but the whole human genome is not possible by gel running method. So during that time, that has been improved, the technique has been improved. And why I told MIT? Because not that MIT is just one of the best engineering institute. No, actually this technique was improvised a lot in MIT itself. And one of the leader there who actually contributed maximum, his name is Eric Lander. So he actually improvised this thing. And Eric Lander did his PhD in mathematics. I'm just telling you because sometimes I listen his lecture, how he actually teaches, because he's very nice, one of the best teacher I have seen actually. Eric Lander, still he is teaching in MIT, biology courses. But I want to tell you, he did his PhD in mathematics, most likely either from Oxford or Cambridge University, England. And then from mathematics, he slowly shifted. And now he's one of the pioneering, I would say, scientists in terms of how DNA sequencing can be improvised in terms of technology, as well as this kind of thing. So you can search in Google that Eric Lander, and you can see some of his lecture, and you will get some, some similarity. Sometimes I see his lecture, he is so good. Sometimes I see how, like, what are the basic point he is covering. So that is very interesting. Okay, so you can take break for 5 minutes, then I will start the next topic and introduce a little bit, because otherwise I cannot finish, because you can understand, right? I go a little bit slow, but this is like my way of teaching. I go a little bit slow, but I want that, I want to explain so that you will understand, and even without reading, you should be fine. Okay. And in addition to that, I have sent you one problem set 2 to 3 days back, right? Have you received it?
Yes, sir.
Yeah.
Yes,
So
sir.
I told you that after this class, because there you have questions from PCR as well as from DNA sequencing. So now you can answer that problem set and send it to that particular email ID. Please don't send me in this IIT KGP email ID, because a lot of emails are coming. So some of them might get, we missed some of those email. So that's why specifically I made that account for your response only. And whenever you will submit your response, you will get feedback from my side. Okay, if something wrong and something needs to be improved, you will get it, right? So please take a few minutes break, and then I will come and start explaining the the next topic little bit. At least we should discuss a little bit. Okay.
Hello, sir.
Yeah.
Sir, I got that assignment from my friend, but I haven't received from you. So is that a problem?
So you have sent your answer already, right?
No, sir.
Send again. Tell me again, please.
Means I got the